Could skin cells one day be turned into sperm and used to create children? It sounds like science fiction, but it is also a real scientific project. The important part is separating what is plausible in research from what is actually possible in medicine today. As of July 2026, there is no clinical evidence that artificially generated human sperm has produced pregnancies or births. That is the starting point, and it has to stay clear all the way through. A big reason this topic gets confusing is that people often bundle together two very different scientific routes. One route works with an existing germline. That means spermatogonial stem cells, or SSCs, and testicular tissue approaches that try to preserve, mature, or regenerate sperm production from cells that are already part of the reproductive lineage. The other route is much more radical. It is often called in vitro gametogenesis, or IVG, using pluripotent stem cells, including induced pluripotent stem cells made from somatic cells, such as skin or blood. That second route is the one behind the headline idea of making sperm from skin cells. Those routes are not just variations on the same thing. They differ in biological realism, risk, and likely timeline. SSC and testicular tissue methods are trying to support or restore a process the body already knows how to do. IVG from skin cells is trying to rebuild that process from a non-reproductive starting point. Public discussion often blurs them together, but scientifically and medically, they are different categories. There is another simplification that causes trouble. Making sperm is not just producing a cell with half the normal amount of DNA. A haploid cell is not automatically a functional sperm. Real sperm production requires correct meiosis, proper chromatin maturation, precise epigenetic programming, acrosome formation, flagellum building, the right mitochondrial arrangement, and ultimately, true developmental competence. In plain terms, the cell must not only look roughly right or test positive for a few markers. It has to behave like real sperm in all the ways that matter for fertilization and healthy development. And that brings us to one of the central bottlenecks, the testicular niche. Sperm do not develop in isolation. Sertoli cells, Leydig cells, and other somatic support systems in the testis help organize and regulate the process. So if a lab reports germ cell markers or even haploid DNA content, that is not enough evidence on its own. Those findings can be interesting and important, but they do not prove correct meiosis, safe chromosome handling, or the emergence of sperm that are actually competent to generate healthy offspring. Meiosis is one of the biggest failure points. Researchers often look at markers such as SYCP3 or GH2AX, and those markers can be suggestive that meiotic-like events are happening. But suggestive is the right word. They do not validate proper synapsis, proper recombination, correct chromosome segregation, or freedom from aneuploidy. In other words, a cell can appear to be moving through the right stages without proving that it has safely done the hard part. A second major bottleneck is imprinting and epigenetic reprogramming. To make a viable gamete, it is not enough to have complete DNA. Parent-specific marks have to be correctly erased and reset. Broader methylation patterns and chromatin architecture also matter. If these systems go wrong, the embryo may still begin developing, yet carry problems that only appear later. This is one reason the field's strongest counterweight is safety biology, not just technical difficulty. Then there is spermatogenesis, the final remodeling of post-meiotic cells into sperm. Again, post-meiotic markers, or haploidy, do not equal functional sperm. The nucleus must condense correctly. Histones must be replaced appropriately by protamines, the acrosome must form, the flagellum has to be built, and the whole cell has to be remodeled into a highly specialized delivery system. Missing any of that can mean the result is sperm-like, not sperm. So what is the strongest evidence that this whole idea is scientifically credible at all? The best support comes from animal models, especially rodents. In mice, a landmark study in 2011 showed that cultured neonatal testis tissue produced spermatids and sperm in vitro, and after microinsemination, embryos developed into viable, fertile offspring. That is strong functional proof of principle. It does not establish full long-term or multi-generational safety, but it does show that in at least one mammalian system, in vitro-generated male germ cells can work. Also, in 2011, another mouse advance supported the SSC route. Haploid post-meiotic cells derived from SSC lines in organ culture contributed to fertile offspring after microinvasive fertilization. Again, this matters because it shows the route based on an existing germline can work, at least in rodents. Later, rodent milestones strengthened the case. By 2015, adult mouse tissue could be pushed to haploidization, though less efficiently than neonatal tissue. Then in 2021, researchers reconstructed much of mouse male germline development from pluripotent stem cells all the way to spermatids, and those cells contributed to offspring through assisted fertilization. That is one of the most important signs that PSC-based work is not fantasy. But it is still in mice, still assisted, and still not the same as a clinical treatment pathway in humans. Rat data adds some breadth. In vitro-produced spermatids in rats also generated offspring by micro-insemination. That suggests the phenomenon is not necessarily mouse-only. Even so, the biological gap from rodents to primates and humans remains large, and the narration has to keep stressing that point. Rodent success does not automatically scale. When we move to primates, the picture changes. Here, SSC-based evidence is stronger than PSC-based IVG. In rhesus monkeys, SSC transplantation after irradiation restored donor-derived functional sperm production, and those sperm fertilized oocytes and formed embryos. That is a serious result in a primate. But notice what it is. Regeneration of an existing germline, not building sperm from skin cells through PSC-based IVG. The more headline-grabbing primate claim came in 2021 rhesus work using pluripotent stem cells. There, embryonic stem cells yielded round spermatid-like cells. After oocyte injection, researchers saw early fertilization events and blastocysts, but there were no offspring. And that limitation matters enormously. Blastocysts are not babies, and spermatid-like cells are not mature sperm. This kind of result can indicate progress, but it does not demonstrate mature, safe, clinically usable sperm. In humans, the most robust achievements remain much earlier in the pathway. Human embryonic stem cells and induced pluripotent stem cells can generate human primordial germ cell-like cells, or HPGCLCs. These are valuable platforms for studying germline specification and epigenetic reprogramming. They help researchers model infertility, developmental biology, and toxic effects. But they are not sperm, and they are not clinically meaningful sperm precursors. Human testicular organoid and reaggregate models have also improved. They can reproduce some tissue architecture, some somatic cell types, and some endocrine functions. But they do not recreate full in vivo testicular dynamics. Human SSC culture remains difficult too. True spermatogonial populations are often lost or overgrown, which tells you how hard it is even to maintain the starting material, never mind complete spermatogenesis. The 2026 human findings were important but limited. One report described round spermatid-like cells from prepubertal biopsies in 3D culture, showing post-meiosis-like markers and even mouse oocyte activation. Another study generated HPGCLCs from patient-derived human-induced pluripotent stem cells. Both are scientifically interesting. Neither demonstrates usable human sperm. And this is the point where careful source criticism matters. In this field, words like spermatid-like, meiotic-like, and haploid can sound more decisive than they are. Marker expression, DNA content, morphology, even early embryo formation after invasive fertilization techniques can all be pieces of evidence without being proof of normal, safe sperm, especially in humans where there are no clinical pregnancies or births from artificially generated sperm. Every claim has to be read with caution. So, could skin cells one day produce children? Scientifically, the answer is that the idea is credible enough to be taken seriously. Animal models show principle. They show that under some conditions, mammalian male germline development can be partially or even substantially reconstructed outside the body, and in rodents that work has led to fertile offspring. But medically, the answer today is still no. Not in humans, not clinically, and not safely demonstrated. The strongest reason for caution is not just that the engineering is difficult. It is that the biology of safe reproduction is unforgiving. Correct meiosis, accurate imprinting, proper chromatin remodeling, complete spermatogenesis, and the support of the testicular niche all have to line up. A result can look promising long before it is trustworthy. That is why the near-term value of this field is likely to be in research, disease modeling, toxicology, and fertility preservation research rather than established reproductive treatment. Artificial sperm is a real scientific project. It is not a human clinical reality. If you enjoyed this, subscribe, enable notifications, and suggest the next topic in the comments.